Hello, everybody. My name is Roman Bauer, and I'm a senior lecturer at the University of Surrey. I'd like to thank the organizers for having me at this conference. So today I'd like to talk about some methods that allow to gain some insight into biological complex systems. And so they go beyond black box machine learning approaches because they allow for mechanistic modeling and or explainable AI methods. So from a fundamental perspective, we can differentiate two approaches that are very different in computational modeling. So on the left-hand side here, you see kind of schematic or a figure that indicates a mechanistic approach, a model-based approach, where we use some kind of a priori knowledge of a complex system in order to create a model and make certain predictions about it. On the other hand, we can also use statistical approaches and data-driven methods that work on usually large-scale data in order to create a model and then make predictions or diagnosis or whatever applications you have in mind with that model. And of course, there are also many hybrid approaches that kind of make use of both to some degree, but these are essentially the fundamental extremes that we can differentiate. And so in this talk, I will talk about both approaches and also indicate or show some kind of advantages of those methods. Now, in terms of biological system modeling, we are using in my lab agent-based models very often, and those agent-based models allow to model biological behaviors as governed by their genetic rules, and these biological behaviors interact with the physical external three-dimensional environment where we have biomechanical processes or chemicals diffusing and so on. And so it is this interaction that is very complex that we can model with agent-based modeling. And importantly, we use agent-based modeling to model how those systems evolve based on a large number of autonomous agents that can be, for example, cell bodies or elements of cells that interact with one another in some way. And so there is not really a global supervisor that tells each agent what it should do. Now, in order to create the software that allows to model such agent-based models and conduct simulations, we created a consortium called the BioDynamo Consortium, which currently incorporates those 8 organizations. And we have worked on various methods and techniques to optimize efficiency and performance of our code. And here, this is one of our early papers where we showed that those kinds of agent-based models can be done in a very efficient manner, and one can gain a lot of efficacy by really thinking about how exactly do you code those agent-based models. And in this case, we had modeled 32 million agents that interact in three-dimensional space with one another. In this case, we have 2 different types of agents shown in orange and blue. The orange cells, they are attracted to one another due to a chemical that diffuses in space, and the same is true for the blue ones. They're attracted to the other blue cells because of a chemical that they secrete. And so then that's why you get this clustering. And so this is kind of like a sanity check that we could really get correct simulations out of this optimized code. And then more recently, we have also published a software called BioDynamo, which is highly efficient and which is actually also open source, so everyone can download and run the code. We have also documentation that is available for free. And so BioDynamo is a software that allows to conduct highly optimized simulations, again, in three-dimensional space by taking into account various physical processes. I'm not going to go into detail here, but we can model really a wide range of physics processes. And so one application of BioDynamo has been on neural tissue development. So we simulate how neurons grow and how they interact with one another and how they create very sophisticated structures in a self-organizing manner. So this is one of the simulations that I think has in the order of a million or so cell elements that can interact. And so one question, for example, that one can then address is, if you look at a single neuron here, can we optimize its morphology? Can we make sure that its morphology matches with what we know about neuronal morphologies from experimental data? And that is the question that we have asked in this recent paper here that we published last year. And so what we did there is that we used machine learning methods and Bayesian optimization in order to calibrate those agent-based modeling parameters in order to reproduce neuronal morphologies. So each neuronal morphology consists of many agents that can be the cell body, and there can be segments of the neurites, of the dendrites and the axons. So this is how the discretization of the agents gives rise to an entire neuronal morphology. And as you can see here, we succeeded in creating neurons that in simulation look fairly realistic. So this is just a visual comparison. We of course did this mathematically. So here you see that this approximate Bayesian computation allows to calibrate those models of neural growth quite well. So in this case, we showed that the calibration with the iterations, the calibration became better and better. And here the dashed line here indicates the actual parameter from a synthetic simulated neuron. And this is what the calibration posterior distribution led to. So you can see that it matches really well. So the brighter the color gets, the more iterations we have conducted. And you can see that each of those parameters gets quite well approximated, even though the model didn't know a priori what those parameters were. So it kind of, based on the evidence, it inferred what those parameters most likely were in a very computationally efficient manner. And we then also applied this to real world or experimentally observed neuronal morphologies. So you can see here that again, we have a clear convergence of those inferred parameters and of the distributions with when we increase the number of iterations. So this is nice to see that this modeling can really help indicate what are potentially the real world modeling parameters that give rise to those neurons. And so here you see some visualizations of simulated and real neurons. So always on the left-hand side, we have the simulated one, and then the experimental ones on the right-hand side. And you can see and you can appreciate that they match fairly well, also visually, not only in terms of various features that we have inferred from those morphologies. And importantly, we have also, we have also used this kind of approach for other applications where we use some kind of machine learning approach or method in combination with agent-based modeling in order to um, to get model parameters that then yield very, very realistic simulations that match with a wide range of different kinds of experimental data. And besides developmental neuroscience, we have also looked, for example, into cancer biomedicine. So we have looked, for example, at lung fibrosis. Here you see a model of the alveoli where in the lung, which make up the lungs tissue, and in patients that have lung cancer that get treatment with radiotherapy, one of the frequent problems is that they can, due to the radiotherapy, get fibrosis of the lung, and this is very dangerous. And so one of the challenges is to identify a treatment regime and the dosage of the radiotherapy that is not too strong, so that um, the healthy cells don't get too much impact from the therapy, but of course the cancer cells um, um, uh, need to get some impact from the radiotherapy. And so we need to find a balance, essentially. That's what the challenge is here. How can we have not too much radiation, but also not too little, and how exactly can we administer the radiotherapy? And so we think that in future, this kind of models can really help optimize the radiotherapy in a patient-specific manner. So it goes into the direction of precision medicine and personalized medicine. And so these are two papers relevant to this topic. Here we had shown that we can actually reproduce fairly well in simulation some responses that people have measured experimentally. And here this is a more general review paper, the second paper here, where we reflect on how one can calibrate and validate such agent-based models, particularly for cancer biomedicine. So this work shows that one can use mechanistic modeling in combination with other machine learning methods, like approximate Bayesian computation, in order to produce and generate mechanistic models that allow to then also produce predictions that then could be in future also experimentally verified. And so I think this is a nice example of showing that one can have model-based approaches in combination with other approaches that are not more data-driven, and still kind of leverage the benefits from both types of approaches and get an explainable model at the end of the day, right? This is, I think, a very important outcome, is that we can create computational models that are not just black box models, that don't just give us some kind of answer without really allowing us to get an insight into how exactly it came to this answer, right? So with this kind of mechanistic approach, we can have explainable AI that allows us to really understand better what are the causes of certain observations. And to go in this direction a bit more, I will afterwards give another example where we used a slightly different approach. And so just to kind of highlight that we are very happy to collaborate with people who are interested in using this kind of agent-based modeling approach. And you're welcome, if you're interested, to have a look at the open source code and download it and have a look at the documentation as well. So as I said, in completely data-driven approaches, we usually cannot really or it's often difficult to get out explainable hypothesis, for example. And one of the previous approaches of data-driven modeling has been in the analysis of retinal fundus images. So the retina is a neural tissue at the rear of the eye, and there's a lot of information that is not only relevant to the eye itself, to the health of the eye, but also systemically about the, for example, cardiovascular risks. And the advantages of retinal image analysis is that one can relatively easily acquire or collect such retinal images. It's very cost efficient, much less costly than, for example, doing an MRI. It's very quick, right? It's a matter of seconds or minutes. It's non-invasive, which is a big advantage. And it's usually not something that's very difficult for patients to do. So there's a high patient compliance. And because of this, one can also do it quite regularly and quite frequently. So one can monitor actually the state of a patient over time. And because of this, people have in the past looked into various approaches to kind of gain diagnostic value from retinal images. So here, this is one of the papers, for example, where people have used deep learning to diagnose certain conditions. However, one problem is that these are usually black box approaches, and so it's very difficult to really use in a clinical setting where we need explainable methods. So what we did is we did some image processing from the retinal images. We used computer vision algorithms in order to, for example, segment the vessels and then classify into arteries and veins. And then we could, based on features extracted from those, from the veins and arteries, we could make certain kinds of diagnosis, and we have published this in the past. And it's not only the blood vessels that we are looking at, it's also by looking at some features of the relevant to the optic disc. So this is the optic disc here in the middle, and one can then have these concentric circles and then have various features that then work with those concentric circles. And so more information on those features that are explainable can be found in those publications here. And then more recently, we also used this to do very performing, very highly accurate diagnosis of Alzheimer's disease detection using inductive logic programming, which is an AI technique that allows for explainability as well. And so here you can see, for example, PyGo, which is such a inductive logic programming method. And you can see here that the accuracy is very close to 100 even. It's over 95% for Alzheimer's disease detection, Parkinson's disease detection, and neurovascular dementia also. So one of the nice things then is also that it really creates explainable hypotheses. So we can really generate certain types of explanations for why the method thinks that, or why the AI comes to a certain diagnosis, right? What is the underlying reason? So it's not a black box algorithm. And this paper has been recently accepted at IEEE conference on AI. And so there we kind of push this approach further to really show that the performance is also very good. It's not only the fact that it's explainable, but we have very good performance. And so to summarize, we can classify computational models into extremes that of course don't cover the entire spectrum, but these are the extremes, into mechanistic or model-based, and then data-driven models. And agent-based modeling is a model-based, is a mechanistic approach, and it can be a very powerful framework to address various questions in biomedicine. And also, we have created a software which is open source, which allows to create powerful computational models, even in combination with black box approaches. So one can combine it and still get an explainable model out of it. And my lab works also on data-driven but explainable AI, where we, for example, look at diagnosis using retinal images. And so if you're interested in this kind of approaches or methods, then I would be very interested to discuss further. So feel free to get in touch. All right, so thanks for your attention. And if you should have any questions, please don't hesitate to let me know. Thank you.